Good morning, everyone. Maria will unmute ourselves. And can we just double check how many people we have, Maria?
Nine,
Great.
including us. Yeah.
All right. Yeah, we have people from far away. Thanks for joining. This is actually a nice setup where we first mingle a little bit and then we have the chance to see a seminar. So the goal was really to guarantee that everybody had the chance to talk to our visitors. And by the formality that this can, be transparent. So we are here to actually exchange ideas and perhaps see how we can all collaborate with our visitors. Is with great pleasure that I am bringing Jonathan's previous supervisor to the lab to visit us. So Dr. Lukáš Spíchal is the group leader of a research institute. In his group is called Phenotyping. So he is at the Czech Advanced Technology and Research Institute at the Phenotyping University in Olomouc. We will have the chance to learn more about where he's from, what he does. So let him do all of that nice talking. I just would like to bring something to your attention. So Dr. Spíchal has a master's in biology and chemistry. He was telling me this morning that he was a high school teacher, or he at least tried to be a high school teacher. I wish he found just a little bit of challenging with that. And then he basically moved on, got his PhD in biology at same university, Phenotyping University in 2005. He does bring a lot of knowledge in multiple fields. What attracted me to his research area was the phenotyping for biostimulants. We do share a lot of common interests. I do think the biggest collaboration opportunity lays in the fact that he does a lot of phenotyping using model plants, particularly Rhabdopsis, and we do a lot of applied research, particularly trying to come up with production methods. So these two together can bring us to great collaboration opportunities. I thank everyone that connected over Zoom to join us. So we'll have his presentation and then we will open for questions after that. Lukáš, the floor is yours.
Welcome. Thank you very much, Randa. Thank you. So, hello everybody again. So it's really, really great to be here. Thank you, Ranito, to have me here. Thank you for accepting also Pavel that is going to spend here 1 month. And Ranito said what is connecting us. Also, there's one important bridge that is Jonathan that is recently working here, but was working for 2 years. It's 2 years, right? 2 years with us. Was working for 2 years in my group. So I think that, what I feel very interesting is that the fact that we do more in the model plants, you do more in the crops. And this is something that I've been told many times, okay, you are working in the model plants, but where is the biological translation to the crops? So we spent some time on that as well. So we were trying to compare the outcomes of using some bioassays, using model plants Arabidopsis, but also some crops. And we do believe that it's possible. Of course, not every time, but for some particular things, you can really find quite straightforward pipeline. So today, basically, I'm here to present the technology. I'm not going to speak about everything we do because that would be for long. I could same time take also presentation not about the technology, so the tools we are using, but I have another presentation ready what we did using or been doing with this technology. And there is a lot about biostimulant. So biostimulant, yes, for us is very interesting thing. I already mentioned during the time before that if we started to put up some high throughput assays, it was mainly because of testing of some simple molecules. So there are people coming from companies with thousands of compounds with the library. So it was more to chemical biology. But then came the biostimulant boom. And as Ronita was telling, yes, it is big interest of the biostimulant domain to get rid of the snake-oils. So I was quite active also in that. There is new European legislatives just coming up. That is, in fact, very much cooperated. The ways are cooperated also with FDA here. So in the new legislatives, the biostimulants will be sold by the producers based on the claims. So not based or fulfilling the claims. So there are 4 basic things that you can claim that your biostimulant is able to do: increase the tolerance to abiotic stress, improve the yield, improve the rhizome. So that's also quite important thing. And the 4th one I forgot just now. But anyway, this is something that you need to claim, and to claim it, you must be able to prove it. So this is why also a lot of methods that we were doing were to use model plants to increase the throughput to do really screening. So so that was also one of the ways of research we did. So let me start. I think it's
Go ahead and click the screen first.
so
Yeah, it's under, the mouse is, you just have to type it out.
okay. Yes, good. So yes, now it's working out. So it's only some bit of introduction. So this is Czech Republic or Czechia. You may remember that we were previously Czechoslovakia. So these are our close neighbors. But of course, we are also neighbor of Germany, Poland, then also Austria, and also close by is Hungary. So I wanted to say proudly what we Czechs brought to the planet, to the mankind. Definitely, some of you are more closer to genetics, or you remember your basic biology lessons. You remember the Gregor Johann Mendel for his inheritance laws that he discovered working with pea plants. So this is something that is coming from our country. Then also we have a Nobel Prize in polarography. This is more into analytical methods. This was kind of forgotten method, but I was told that these days this is again coming back a bit. So we have the Nobel Prize winner Jaroslav Jerovský in this method. Then the word robot. We use a lot of robotization, robots. Robot was introduced as a new word by Karel Čapek, that is a novel writer. He wrote before the Second World War. He wrote a lot of also, we call it maybe today sci-fi literature. So he introduced robot. And this is why, or why it is so. Robot is coming from ancient Slavic word robota. Robota describes like a hard work where simply you are only working and only working when people were not free, when people had to do what somebody was telling them, so it was robota. So something that you do all the time, manually, really tough times. So this is why robots. And I think it was right, because what are doing robots now? Robots are trying to substitute people in some very routinary operations, right? So then many of you are probably also using contact lenses. That's our invention. Unfortunately, we were at the time a communistic country, and it was not allowed to be wealthy. So Professor Richterle was the organic chemist that developed this material, and he produced the first contact lenses. Our country was so stupid not to allow him to yield from the patent. So the patent was sold very cheap to Bausch and Lomb. So, so that's how it goes sometimes. And I think many of you are drinking Pilsner. Pilsner is called because of the technology, how you brew a beer from a place called Pilsen. So Pilsner beer was introduced to the world here. So you also maybe know Pilsner Urquell, that is the real Czech beer, the first Pilsner beer that appeared in the world. Anyway, so coming closer to my town, so it's Olomouc, as was mentioned, and we are proud to say that it was our student. So we studied for 3 years at the university, Mendel. We gave him good base, good foundation. Then he moved to Brno and then to Vienna. So there he made his main work. So people from Vienna or from Brno, they are probably telling Mendel, this is our boy. I'm always correcting them. Yes, he did well because we formed him well. So anyway, the education in our city is very important. We are 450 years old university. Just this year we are celebrating this. And Olomouc is a really beautiful city. So if you have a chance, don't go only to Prague. You can have less tourists and very similar things also in Olomouc. It's a very baroque city. It's called also Small Prague. So people that are visiting our country and that are coming to Olomouc say, oh, wow, this is like combination of Prague, Vienna, and Krakow in Poland. So a nice place to be. So feel free to come. We'll be happy to see you. About our group, as Vanito mentioned, we are in a part of a new built institute within our university that is called Czech Advanced Technology and Research Institute. Our university, short from Catherine. So we use Catherine as our nickname. I am group leader of one of the groups there. We have there about 20 groups, and our group is called Phenotyping. So if you are interested to know more, you can visit the Catherine webpage. And then it will lead you also to my group. We are about 10 people. We are quite interdisciplinary. We're speaking here about it, how important it is. So we are only missing mechanical engineers and IT guys. So that's the problem because our university is a very classical one. So we don't have any technical study programs, unfortunately.
So you cannot talk to George.
I just said something. Sorry. Anyway, these are our training, this is our profession, but you see that we also have some needs. So it's already Pavel was introducing himself. He's analytical chemist by training, but these days he's more hardware software developer. And also more and more we are turning into data analysis. So for example, a very important part of a member of our group, Nuria Veriego, she's plant physiologist, but mainly because she's plant physiologist with also agronomy background. She's the one that is now, in fact, more and more working on the data. We were speaking about it. So the more and more data you have, more and more headache you can get as well. So that's the problem. So this is who we are. We do also not only phenotyping, we do more things. We have a lot of interests. I am interested in many things. So I'm making headaches of my people coming to the seminars, telling them, and you know what? I was thinking this. But I have very responsive people that are able to tell me, well, slow down, and that's it. So we are interested in all these types of phenotyping. We already spoke about biostimulants. So this is, let's say, our very core interest. We have also some basic research running that, in fact, developed from the biostimulant research, that is polyamine research. So in cooperation with people in Mexico, especially, there is one group that we are very much collaborating with. We study the new aspects of polyamine biology, that is transport, is metabolism. So we generate a lot of mutants and are trying through phenotyping and interactions with the environment to study their importance. We start to be also, and again, it's connected to biostimulants, interested in bio-waste valorization. So it's also kind of field of interest. And why I'm here and Pavel is here is, of course, we want to find also use of these technologies somewhere where we believe can be translated very easy. So this is, for example, a CEA that you are experts here in. So now I will only shortly speak about the phenotype-genotype environment interaction. This is something you know perfectly. You can have one seed batch, you put the seeds in to grow in different environments, and you get completely different plants. So that's the very simple thing, that you have interaction of environment that is and the genotype. So in fact, the environment is pushing the genotype to evolve into different phenotypes. So this is what we study. So of course, if you have this, or I use this visualization like a basic principle that basically if you sorry, there's something missing. No, there is no. This is later coming. The important aspect is that the phenotype plants is changing, is dynamic. So the aspect of time, the fourth dimension, is very important for us. And the importance, of course, then, or how we can use this advantage through using non-invasive methods. So you simply follow all the life of the plant, and you can change the environments, and you can see how it's changing the phenotypic plasticity. So that's very important aspect. So of course, what is happening? There are several factors that are pushing on the plants. So there is, of course, the environmental factors, some biotic and abiotic stresses that are changing the phenotypes different ways. And then you have several types of technologies that you are trying to push back. So there's a lot of breeding or GMO or CRISPR-Cas is these days a very powerful thing. GMO, big problem in the European Union. We are simply, old Mother Europe, simply very slowly moving, very elegant, but very slow. So there's a lot of fight also to change legislation to allow CRISPR-Cas as a non-transgenic approach and to accept it as a way how to do things. Of course, there's also many other things that basically we are more interested in. That can be agrochemicals, that can be biostimulants, through which you are trying to help the plant to cope with the environment. And as I mentioned already, the important thing is the phenotype plasticity. So mainly we are not interested in some endpoint situations. We want to see what is happening over the time. So what we do is we are monitoring the environmental interactions of plants through using different types of assays at the different levels. So we use some high throughput assays that are, let's say, rather bioassays, where we use the advantage of in vitro or semi in vitro. That is kind of strange to say, but maybe then later I can explain what I mean by that. We use the whole plant assays. That's also very important. You want to see the response, including when you are doing screenings. You want to see the response at the whole plant level. And then, of course, we can do also high precision phenotyping when we are using not simple readouts, but we are using very complex way, using more sensors together, and then trying to really integrate all the information. So there, of course, the throughput goes down. So the keywords for us are then, of course, phenotypes to be non-invasive. At the same time, of course, we can complement our research with some invasive methods, sampling, doing some analysis. Biosync automation, very important, and then either high throughput or precision. And everything you will see here is mainly in controlled conditions. Okay, so this is controlled conditions. This is not phenotyping in the fields. We are not doing that recently. So over the time, we simply try to put together something we call Olophen. It's not very inventive. It's Olomos and phenotyping. So it's a platform. If you Google it, you probably find it as one of the platforms that are on the map of the International Plant Phenotyping Network. And yeah, there are already a lot of platforms all around the world, specifically in Europe. It's really dense these days. So we are part of it. We do a lot of things simply to find new technologies for sustainable agriculture. This is also a fancy way to say what we do. And you see that we can combine several things, but what is very important through trying to implement such things, we found out that we really need to do in higher throughput, or that the higher throughput is coming naturally when you want to really look around the plant. For this, you need to use automation to some extent. And we are using the non-invasive method. So the Olophen platform using phenotyping technologies that I will present today. And for your navigation, you will see these colored dots that are telling you if it is high throughput, high precision, or affordable. phenotyping technology, but you will see that also these things can be combined. So it's not only this thing, but there can be, so this can be high throughput, high precision, including it can be high throughput affordable. You may think why there are these two blue, what does it mean high throughput and high throughput? This is the way I decide to say ultra high throughput. Okay, so I'm more and more, of course, trying to do marketing of the science. So I'm developing also new interesting names for what we do. So following the analytical chemistry, where you went from basic HPLC to a high throughput HPLC, or how do you call it, high precision, high performance.
Ultra.
Then now you are in the ultra. So I like it. So this is the ultra high phenotyping. So now I will show you like a 1-minute video that is kind of a tour through our facility. So you will see it in your own eyes. So let me start it. So these are the buildings. You see, we use also some robots. So this is Cinderella, that is simply handling all the seats pretty fast, pretty efficient, saving a lot of sorrows of technicians. This was done in collaboration with a company that borrowed us this machine. It's one of the invention of Pavel, by the way. And of course, we move into some laboratory conditions, closed chamber conditions. We then go into the whole plant analysis. So we are using this type of closed chambers, big closed chambers, where we have this automation. So yeah, as you will see later on. So we have the top view sensors moving positions. This is for the high throughput. This is for high precision. Here you see that the plants are going to the sensors, and they are simply analyzed for different things. And then, of course, it's the analysis. So this was kind of this was done, the video was done by the company, and this is Stoigli Robotics that made it for us simply to have some promotion. So we are thankful to them. So what you could see, one of the things you could see is this so-called top view imaging XYZ system. So here is the render when it was designed. Here's the reality. So you already saw in the movie that we have this growth chamber where we can control the conditions fully. So we control the light. We can go up to very high intensities, including 1,000 micromoles per square meter per second. So we can really go to super high intensities. We have cool white there supplemented with infra. So partially we can play with the quality, but nothing serious. We control the humidity, we control the temperature. These chambers were designed to have really as less microclimate problems as possible. So we did a lot of really standard-based analysis, how homogeneous is the place, and it is. So we have this robotic arm that holds 3 types of sensors. Is the RGB classical camera. We have there also the floor cam unit, so we can analyze the kinetic properties of fluorescence of chlorophyll. So there we can extract a lot of parameters connected to photosynthesis efficiency. And also we have here the hyperspectral unit that works in the visible range. So NDVI, pigments, and this kind of things we can do in the same time. So the good thing is that this has quite nice capacity. So these trays that you can see recently there can be, I think, 64 of those trays. Speaking about a plant, model plant Arabidopsis, when it is simply adult plant, we can have here more than 1,200 plants for one screening. So including for lettuce, this can be close to 1,000 plants per one run. The very important thing is that here we can only analyze top view. Yeah, so this is for the horizontally grown plants specifically, but we use it more for screening approaches. So we miniaturize the whole system into the multi-well plates. So here we can place more than 500 plates, and basically now we use the plates that have 48 wells. So then you will see the multiplication. You got over 25,000 plants per one run. And the run takes 1 week, so it's pretty powerful tool then. We use this thing a lot. I will show you later on. The second system we have that you could see, if the previous one was so-called sensor to plant system, this one is plant to sensor. So we have this growing area. From this growing area, you have the conveyor belt that is simply taking the plants inside the system. You can do dark adaptation, you can do automatic weighing, you can do automated irrigation there. So there's a lot of things that can be very complex. The throughput is going down here because we have also possibility to see from the side view, at least for RGB. But also we have here more cameras. We have also thermal cameras, so we can do the thermal imaging. And we have another type of hyperspec that is for the short wave infrared. That is very interesting tool. We still have some time to work with it, to use it better. But there you can analyze in an invasive way the amount of water in a plant. So you get an absolute amount of water in a plant in that moment. So when you combine it with thermal imaging, that is basically telling you about how the stomata are opening, then you can also get a lot of information about how the plant is performing in the sense of water management. So then combining with water stress and heat stress way of experiments, it's quite interesting to have. So we are using, these are simply the toys, they are the tools. You need to use them for something. So the sources of of the biological activity on plants to analyze used to be genotypes. So we work also with people preparing a lot of the types of mutants or breeding materials. They are interested in testing it in different environments. We analyze compounds, so it can be libraries of compounds. I mentioned biostimulants. We can also with microbes. So we do a lot priming with microbes. So that's quite an interesting thing because what I believe, what biostimulants are really having future are those ones that are connected to the microbial activity. So that's something that's in our opinion and experience is something that really works more than some extracts from seaweeds, for example, etc., that are working for sure, but in some really only in some scenarios. So we published some works doing phenotyping after exposure to volatile compounds. They're also quite interesting because volatile compounds, of course, are the messengers, how the plants are somehow communicating within one plant, but also among the plants. So all this we can do in collections, libraries, batches, series, etc. So that's the high throughput. You have a lot of things that you want to analyze. How we apply through seed treatment. Companies, they are very much interested in seed treatment. So it's priming basically of the seeds with some compounds. Then we are, of course, able to do some foliar treatments. I don't have pictures, but we don't use a lot of this, but we have also some spraying cabinet where we can precisely spray the plants. And of course, you can apply through drenching. So the assays we are using are, let's say, in vitro-based bioassays for hormonal response. We analyze the seed germination, shoot growth response, and the whole plant assays where we again are trying to get focused more into some events of developmental stages like emergence, cell development, and overall growth and physiology. And we use so-called normal conditions, so simply whatever you can establish as a preferred conditions of growing of the species you are analyzing. Abiotic stress for us means salt stress, drought stress, heat stress. We are becoming a bit bored with that, so we are starting to combine the stresses to have more complexity, and I'm including somehow finishing with that. So we are going more and more into biotic stress, and we already are close to publish some high throughput methods for analysis of the biotic interactions. It is in the model plant, this is the pathosystem, is Arabidopsis pseudomonas, and we have also botrytis to work with. So if you are interested in some pests and studies of this, you can also address us with that. So the future definitely for us is then not only to do combination of different abiotic stresses, but combinations of biotic and abiotic stresses. Because this is what I believe that is really happening then in the environment. You never have only one factor. And recently it's also shown that also some some pests, for example, aphids, they don't like a lot to infest the plants that are suffering drought. And why? Because of course the plants, they are changing their metabolomics, they are changing the components, they are less tasty maybe, right? So this is quite interesting also into some, let's say, the protocols, the practices, managements, when you can really prime maybe the plants to let them suffer with a mild stress that still is not like giving you a lot of yield penalty, but you can make those plants more tolerant to some kind of other stresses you are so far not thinking. Of course, you are in CA, so you say, okay, I'm in a closed environment, I can get rid of it quite easy, but well, still can be quite interesting. So all the things that I already mentioned, we do in kind of pipeline. We already spoke about some metabolomic profiling. So this is like a targeted way to do. Of course, we also, when people are interested, also in inspection of some gene expressions, we can sample the things. And now doing in a targeted way, we have some batteries of genes that we know that is worth to analyze. Or of course, not in our group, but in close collaboration with the next group, we can do also RNA-seq to really have very detailed inspection on what is happening. And of course, all this is not always coming linear way. We are not for every experiment using everything, but that's the ideal situation, okay, that if we decide to do something very strong. So now let me lead you through some examples. So the really high throughput is running in the multi-well plates. What is important here, this is fast, okay? So you are in several days, you have the information of many samples. The throughput here means not only a lot of different things, but also a lot of variants of one thing. So here we are trying to do time series. We are, of course, having a higher number of combinations. So speaking about application of a chemical, to apply it in one concentration, you can miss the hit very easy. So we go over quite broad concentration range. Speaking of biostimulants, we start with 1% dilution going to 0.001. Sometimes we are surprised where is the window. The windows are also moving with different stress that is coming into play. So this is why there our throughput is needed, or higher throughput is needed, because we really don't want to miss the hit. Then, of course, very importantly, higher number of replicates, more robust statistics you can do. So you can really see statistically significant differences, very small things like 2% of a rosette, for example, size. We are able to track. And if you have more plants in your trial, you can also, or I think ecologists probably will not agree with me, so I'm simplifying a bit the situation, but I'm speaking about the population behavior. So if we, for example, in one essay that I will show, we have 110 seeds and we are looking at emergence as a readout, then it is 100 seeds from one batch. So it's already giving you some information about heterogeneity or homogeneity side. So this is quite interesting aspect. So all this we do in the microwell plates. So that's something that is quite ready for some screenings. And importantly, this is our, let's say, very simple but very successful. Maybe because it's simple and successful. A situation or assay that we are using 40 egg well plate and Arabidopsis plant growing in that, and simply analysis of the projected area. This is giving us a lot of traits of interest. So it started in 2017 when we published this paper where we claimed that this can be really interesting way to do the screenings for let's say discovery of potentially bioactive compounds in wide range of concentrations or genotypes under various growth conditions. We went further. We improved that. This was only in a not very high resolution, but we went 1 year after. We had very good PhD student, Lydia Opena from Spain, that PhD with us. Now she's very successful doing career. She's an R&D coordinator in Trade Corporation that is quite successful, worldwide successful biostimulant producer. So we are happy we gave her training because she's coming back with some contracts, of course, because she knows perfectly what she was doing, and she knows now perfectly how she can use it. So instead of establishing it in the company, in her own R&D, she's simply using this opportunity, and we are cheaper as well. That's our opportunity as somehow. So what we did here, we improved really well the throughput, and as I said, in 7 days we can analyze more than 25,000 plants. Important thing is that it is not only about individuals, but this is always 48 plants per variant. So speaking of let's say one trial, you can have more than 500 variants of one trial. So that's quite cool. I mean, when you start to multiply, then of course it's not so big deal, because starting with 3 concentrations of a compound in 4 different stress situations. Every stress situation, if it is a chemical-driven stress, then you can set up in also concentration dependent. So salt stress. Okay, so let's have 3 levels of salt stress. So you have 3 pair 4 pair 3 again. So then you are really seeing that this is not so much, but still it's cool. I mean, yes, we are proud of it. So definitely, this is something that is then this double dot, ultras. But well, I mean, how we do? So we basically take a top view RGB picture. Not big deal, because we have the control conditions. So we have the light conditions set up. Important thing, because if you want to do the RGB analysis, you need to have the RGB well set up. So twice a day we take a picture. So the one picture is, Pavel, please correct me if I'm telling something wrong, because you are better in that than me. I'm only claiming that I understand. You do understand. So this is 10 megabytes of a picture. Okay, so 10 megabytes of data you are taking in one shot. So it's 20 per 1 plate a day. So then in 1 week, for each plate, you have 140 megabytes. But you do more than 500 plates a week. So then you end up with these numbers. 7 gigabytes. Let's think of 50 working weeks. Okay, if we go really to the push, okay, push. Then in 50 weeks, this assay is giving us almost half terabyte data. So that is the new bottleneck for people like us that normally don't have supercomputers. But mainly, it's a lot of data to process, to do image analysis. When you do image analysis, a lot of data, because you only turn the pictures into the zeros and ones. So into some numbers. Then you need to do something with the numbers. So it's another challenge. So this is why we are happy that Pavel is a member of our group, because we simply, in fact, he came along with that this RGB-based, color-based analysis are his slides, so we can then tell you more. Simple RGB is not enough. A lot of mistakes, simply accuracy very low. So we try to improve it through using the UE saturation value way of analysis of RGB. So he was playing a bit with the segmentation using these changes. So like on the mixing pult, you were trying to say what is the green. So what happened was that it improved really something. We had really good things, but we had a lot of problems. Small plants. Big problem, sometimes recognizing, sometimes not. Because remember that we are also applying the stress. So the green is not the constant green. It's changing within the time. So another challenge, you have some reflections. So these reflections are giving a lot of mistakes. So, well, Pavel said, this machine learning and stuff, so I can do something. I said, good, go ahead. So he did. So he used, through some Googling and tutorials, he simply came with this UNet. UNet, Wikipedia told me that it was discovered for some biomedicinal research. So it's a machine learning method based on the convolutional neural networks. And he applied this already existing approach, and he trained the model using only 500 pictures. You remember how many pictures I showed you we are able to generate a week. So it was really like nothing. And already with this 500 pictures, he finished with accuracy of 97%. That was really cool. And of course, I don't know where we are now. If you had time to go on with improving the accuracy.
Actually, no.
Okay, but 99.9, I feel like it can be really cool. So we will see. Then this is the important outcome. Only using this, we really improved the method very significantly. So this way, we have no problem with the small guys because the color doesn't matter a lot. It's trained to recognize them, and machine learning simply decided what is the approach to do. We don't know. Still, we have some problems with some reflections, but again, it's about a retraining of the model. So I believe that if you retrain the model with many pictures showing this is not the thing, then or having a lot of pictures like that telling not this is not the plant, then definitely we can end up very easy with that. So that's our experience, basic experience with using deep learning. And I'm very happy that it's not such a big deal. I don't know what is your experience. Maybe this is like very basic for you, but we are very happy with that. It works. It does work. So very importantly, having this very fast and automated way. Jonathan can tell you what is then to use the already commercially available softwares that are able to segment. But it's still a lot of work to feed the program with the pictures, right? To check if your mask is working, if the mask is well set up. This is working simply immediately. So that's cool. And this is allowing us then to read a lot of things out of these pictures. Area, perimeter, RGB color index. That's for us is like a stress response readout. But well, as I said, this is cool. You get a lot of Excel files or CVS files with a lot of numbers. So what does it mean? Then somebody needs to sit to the computer and start to work. That became another bottleneck. So again, we are so grateful to have Pavel that is so open-minded. So he said, okay, so what if, yes, I don't care what is after the if, yeah? Yes. So what he did, he simply prepared again some Python script that is able to take all these numbers that are in the Excel file or CVS file, wherever it is, and turn it directly into the outcome. So directly we can take some traits of interest plus visualization. So you get graphs for all the kinetics of the growth. Yeah? So all these exponential or different behaving curves of your variants, how they were growing. Okay? So this is each point is the projected area. Yeah, so over 1 week, you've got these 14 numbers that are giving you such a nice curve, and we can do the analysis on the curves. That's the need of the using of the statistically or the advanced statistical methods, because you are doing really the statistics on curves, not on points. So another direct outcome that you get as a package, you get a package with the graphs. So it's perfect. It's standardized. It's unified. Because I'm telling you that each of you, if you go through a basic training, and I gave you the files with those numbers and ask you to prepare outcomes showing me these graphs of different traits, there will be big diversity in your outcomes. Yeah, so we wanted to avoid mistakes. Human mistakes can happen, of course, it's normal. We wanted to avoid also, let's say, some freedom of decision of a man, because somebody is more familiar with more physiological traits, somebody is less familiar, somebody feels like, oh, this is more important for my research. No way. First, we must finish with a standardized outcome. So standardized outcome, some distribution values, parameters in the groups. So this is something that I was calling like the population behaviors. You can have different plots of endpoint or single point traits. We have very important thing that we are using this spiral coordination plot. That is something like a visualization of all the traits that we are analyzing. So you see there is 1, 2, 3, 4, 5, 6, 7, 8, 9 traits we can read out from these simple pictures of the Arabidopsis taken over 1 week. But then again, it's how to have for the first glance, what is similar to what? The 500s of your things. So then we have something like this, and I'm calling it like a phenotypic fingerprint. This is simply like a first information for you if something behaves the same or different. So you can very easily then go to some parts of your trial, say, aha, here this was the information popping up. So this is very important part because it is telling you how they are doing similar or different. This is normalization. I will explain it later on. Because the very last outcome and the very first to see is in fact a heat map. But how to do from something like that only one number that is then giving the color. Already this is a normalization. So this is log 2 simply is the blue dashed line is a control, and you see how the trait is changes plus or minus, simply simulation reduction in each of the traits. So we simply sum it up and we finished with one number. So then you can have all these 500 experiments expressed by 500 numbers. This is then giving you really like the first clear overview, what you can see and what you are not seeing. So you say, here is something that is becoming positive. So let's go reverse. So let's go into here. What is the main component of this blue that is positive? Okay, so it's this trait. So let's go deep into this trait. And then you opening backwards and you get into the Excel file and you can select the information. But you are starting with a standardized outcome. So this is like a very short way set where we came after 5 years. And I'm so happy for that, because really it's something you also for the training. If you come over to our lab, we don't need to give you really a lot of training. You will do the experiment and then you will come with this to the seminar and say, look, I got this. Let's discuss what does it mean. So we are really shortening. So this is another sense of the high throughput for us. So let's jump from the Arabidopsis. Using the control environment, I know it's a lot of pros, but also a lot of cons. So one of the cons is the limitation of the space, limitation of the conditions. So for us, the limitation of the space is then the size of the plants. If we want to improve the throughput, we cannot work with big plants. So we go into the same plants when they are still small. So here, the time of emergence of the plants was for us the important time. It was the trait we were following. So what we do here, we simply take, as I said, 110 seeds. We put them into the soil. So importantly, also this is soil-based assay. And every second hour, we are taking picture of the trait. And you would say, okay, this is the same seed batch, so they all should be the same. But no, of course, you have really dynamics over the time. So this is the time, and this is the number of emerging seedlings. For us, emergence is simply the time when we see the first recognizable green pixel. So it's not a physiological emergence, let's say theoretical emergence for us. But you see that basically this one are the fastest one. These are the lazy ones. But you see that you can have some nice dynamics when you apply stress. The slope of the curve is changing. So this is very interesting. So you can then do some mathematical operations to have at least 3 traits that are telling you how fast it happened, that the first or the last appeared, what was the time between the first one and the last one, and also what is dynamics so this is the dynamics, and also what was the final number. So this is the time to event analysis. So we are not putting endpoint, but we are simply waiting until all are done. And this is then easy to quantify as well. So still here we are able to do 60 variants, so quite interesting. This, of course, is mainly for the technologies that are applied to the seeds. So very much interested for the seed companies, for the agrochem companies. we are doing this in the stress. So as you see, the stress is simply really clearly influencing the emergence. It is desynchronizing the population. As another, so this was already still high throughput, but using some crops. So you can do it in whatever crop you can imagine. Of course, yeah, depends on the size of the seed or not. Depends, but we were from the poppy seed that is really tiny to the beans, very big ones. So it's quite simple and quite powerful. To show you something that can be high throughput or still large scale and high precision. We also published this paper when we went into cereals. This was done with barley. That is important crop for us, beer brewing, of course. And there's also a lot of interest in breeding of barley. So here we used very simple thing, combination of the side view and top view, and also fluorescence imaging. Here we combined also with the metabolomics. So this was very nice work that was finishing with a lot of correlations. And yeah, this was again quite interesting to see that the simple side view of a canopy height development was a proxy of the tolerance of the plants to the stress and of the recovery to the stress. This was a water limitation. So this is what you see here. So the simply analyzing in time the average or mean height of this canopy. So it was not with simple plants, but with canopies. Then you can see this each point. So this is where it's well watered. Here is coming the water stress. So the well water is continuing in its height, but here it's like decreasing. Yes, it's losing the turgor, so it's starting to bend, and you have like they are decreasing in the height. When you then rewater, you see that again you can have a very nice recovery, and again very easy mathematical operations looking at the slope of the curves, slope of this curve, slope of this curve, and slope of this curve is very interesting then proxy of how the plants are coping with the stress and recovering from the stress. Our, let's say, so far best published high precision result is this one that we published quite recently in Plant Journal together with people from Poland. They were interested in finding some, again, metabolomic markers of the mild stress response. They used pea plants. Because of the classical physiology, pea plants are still for physiologists good plants to work with. So we used our facility to analyze not only RGB but also infrared, so the thermal imaging and floor chem. So we did a series of the observations. We quantify a lot of things, and we were able to then, together with the combination with metabolomics, to analyze really some markers that can be already like indicators of some stress, water stress coming. So that's the orange dot. And then this is Pavel. Sorry, Pavel, I didn't tell you that I'm going to show you here, but I knew that you will be anyway here. So that's something that we developed to be more affordable. Because many people were coming or still are coming to our facility and saying, oh, wow, but this was expensive. No, I said, oh, it was, yes. We had opportunity, grand opportunity, so we simply could afford it, but it's not really easy. It's a lot of money to have it, but a lot of money to run it as well. So we were thinking of how to change the thing that is horizontal into simply to have less space. So the same, like if you grow your strawberries in a horizontal way or in vertical way, you simply are improving your traits per square meter. We did the same. So we got some internal grant at our university. It was pretty easy because it was grant opportunity for like a new technologies. There are not many people at our university that are dealing with technology. So I talked to Pavel and his close collaborator, that's also Pavel. So I talked to Pavel and Pavel, well, guys, so there are some money on the table. So come on, let's go and take it. So I told them what is my dream and where are the money. So they made the dream true with those money. So this is something that we call vertical phenotyper. And it's simply stacking the things in the space. So we have these racks Here are the multiple well plates that are moving in the space that way. So you are perfectly using all the volume only on a footprint of 1 euro pallet. I don't know if euro pallet is good metrics here, but how much it is, like 1 point what square meter?
1.2 per 80 centimeters.
Okay, so let's say about 1 square meter, something like that. So this, sorry,
3 by 4 feet. I understand
so
that.
it makes it very understandable. So it's good. Thank you very much, Gonzo. That is very important. So on such a footprint, we were able to place something that otherwise was on 2.5 square meters or maybe more. So that is very big advantage. The good thing is that this chamber is in fact growth chamber itself. So we can control the light, we can control humidity, and to some extent, of course, the prototype still. We know that there's a lot of space for improvement, but importantly, we have here simple Raspberry Pi cameras that are many there. How many? 32?
28.
28 cameras that are allowing to take picture of every object from top, from bottom, from one side and from the other side. So we are improving this way also the potential to take more information. So this way also another thing you have here is 3D printer as well. Pavel has 3D printers and he's busy with prototyping also some accessories. So we are trying also now to show that because many people are asking us, do you do also root phenotyping? I say no, and now I can say yes. Because this basic root phenotyping, when you do the small seedlings, typically in Arabidopsis research, you do a lot of this stuff on the vertical plates. So this we can do now using this thing. This is going to be commercialized, and we hope that it can be of interest. Because of course, along with this is not only hardware, but there can also come the software with that. That is giving you all these packages that you could see we developed for our own research. So this is kind of like to also commercialize what we did for the research. Then we have another thing. So this can be of your particular interest because this is simply array of 40 Raspberry Pi cameras that are in place in this grid in a growth chamber. It can be in the greenhouse as well. And here we are simply taking the opportunity of the Raspberry Pi being cheap, easy to work with, or in my opinion, or I don't have to. So okay, easy. Let's stick with this. So anyway, we can have here 40 of our standard trays, where we can then let them sit under the camera. The very important thing is that we have also some stabilization of the light. So every time we are taking a picture, we are overflashing the light, the light that is otherwise there with supplemented LED stripes. Yeah. So this way we are trying to homogenize the condition to have easier to work with the pictures. And here is also a lot of room for the machine learning. So we are now working on that, especially in this emergence assay, that also for the ITI became very interesting because they said, okay, here we need to work not with one point, because the simple training of the network telling now this is the emergence, it was not very accurate. Why? Because there's a substrate. So the background is then giving a lot of troubles. The background can have some particles that can look like a green pixel. They can be, of course, the background is also changing the color, because if you want to have a well-watered substrate, then it's dark. When you let it dry out, it's turning into brown, yellow, something like that. So this is the complication. So the IT guys working with us were quite happy with it, because they could, of course, do something more complex. So now they are doing some more elaborate way of machine learning, working not with the simple points, but with the data rows. They have simply several pictures before the event and several pictures after the event, and they are playing with this, or they are training the model using this approach. So the good thing on this is that every picture, when it's done, goes directly into Dropbox, I believe, or somewhere simply into some data storage. Of course, it's very simple, but very useful. So as this thing that we developed for a bachelor student that was trying to grow hydroponically some basil plants. She had like 100 plants, and I was torturing her with the request to have every day some data. So Pavel was again close, so he helped her a lot because he constructed this very simple box where you have camera here and camera here, and you have here a table that can be simply lifted, so you can move it or change the position by or change the angle by 90 degrees. And this way, again, nothing very much sophisticated. Simply the student took the plant, put it inside the box that was already in the greenhouse, so just close by, and touch the button. In that moment, one picture from top, one picture from side. Then simply moving position by 90 degrees. Again, push the button, and you have again top and lifted side view for some analysis. And then, of course, she used only the software that we have for this, let's say, morphological analysis, and she finished with very simple experiment with a lot of data. So a lot of data that then, of course, changed her thinking of what she's going to do. She was thinking that she's going to grow basil in a greenhouse. At the end, she was sitting at the computer working with data. But yeah, I mean, this is very powerful thing. So easy. So this is what we do to be more affordable, and that's it. And you see that over the time there were many people that were going through our group. So you see that there are not only Czech-looking surnames, but also a lot of people coming from Spain, from Mexico, from Latin America, yeah, from also some other parts of Europe. So if you are interested in whatever, you can ask now, you can ask later, or you can simply send me email. I really, if you are interested in very detailed things, use your time when Pavel is here. And he came here not only to learn things, but also to help. Okay? We love to share. We love to share, I believe, and I'm saying this because I love to share. So this is the tool to share. Well, thank you for attention.
Just before we jump into questions, this is an important clarification. So Lukáš will be here for 1 week, so that he will depart on Friday morning. If you have the chance and want to talk further.
This Friday?
This Friday.
So it's less.
It is,
Yeah.
it is, yes. This Friday.
This working week.
No, even.
Well noted.
Yeah.
And then Pavel will be here for 4 weeks. It's nice that you have 2 Pavels, right? So it's so good that you cloned him.
You have to keep one.
Yes.
So maybe we can start with the people connected over Zoom. If you have any questions, now is a great time for you to ask. And then we go over questions here.
You can fill them. It's
No questions? Online? Oh, we have, let me see. No questions at the moment. So then go ahead, please ask whatever you want to Lukáš.
Yes, please.
My question is about the application more than the system, because I love the high throughput system. Have you ever looked at genetic variation in response to some of these things, especially in response to, say, biologicals and such?
Mhm. Yeah. Yes and no. The point is that this type of work is not our interest, the core interest, but we are very much open to people to come with this problem, because what I say, speaking about variants, of course, this can be different genotypes. So, for example, what we did some phenotyping with using some, I don't know, about 100 accessions of Arabidopsis or some GWAS. So it was Plasmodiophora treated 100 accessions of Arabidopsis that had to be used for some GWAS analysis then. We'd love to help also the genetics, but somehow I was not able to attract them, or I haven't been able so far. So that's something that is in fact for us super easy, because then okay, if we have the seed batches, then simply we can spread them and analyze. We can in fact it's more simple than we otherwise do, because we don't need to add so many things, because normally if we are applying agrochemicals or plant growth regulators, biostimulants, and of course in the protocol is quite complex. So there's a lot of variability of the protocols because of what you want to add. But if you have the genotypes, you don't need to add anything. You have it already in there. So yes, that would be perfect, but I don't have any clear example what we could do so far. But it's ready for that. It's ready for that. Yeah. 500 variants.
This is obviously a very expensive research. What are the main sources of funding? You indicated some funding was provided by the university, and you mentioned one of your former students is now working in the private sector and kind of collaborating. Get funding from the European Union.
That's a very good question. Yeah, I mean, this is where we need money, to run the experiment. So you're right. This is very, as I said, it's not only to get it, it's to run it, and we had to find a model how to be, let's say, independent of several situations that can happen, and they are happening. So briefly. We were lucky to get it because there was the time, I think 2003, Czech Republic entered European Union. So the European Union is having some structural funds to improve the infrastructure in the country. So the buildings were built. There was some money left. Mr. Director was asking, okay, we have quite some money that we spent in infrastructure, or we must put back to the government. So who thinks we can use it and how? I was the fastest, more convincing, so we got it. But then started the fight, because when we got it, I went to the director. I said, "So cool, we have it operational. Now I need 3 people."
Find them.
Good for you.
If he says find them, I said I have them.
Right. That was pain.
Yeah. So that was really big problem. So in fact, I had to use some national grants. We have in our granting scheme in Czech Republic, we have basically 2 main important grant agencies, one for basic research and one for applied research. So yeah, sometimes you must improvise. So the starting of the operation of this, that comes back to 2013. I was using money that were dedicated to different things, in fact, to be able to take people to work there. Then came some other, let's say, the continuation of the structural funds was also to keep something that you built. So there already I could have some dedicated money, but from the beginning, having this, we started to offer this also for contractual research. So there were companies interested in using that. Took some time to introduce this to them, because this was completely new. I'm speaking about 10 years ago, and they were like, "Okay, it's not possible." So I said, "Like this, come and see." So they were, of course, able to pay. And the other problem coming with this is then you cannot do only contractual research, because this way you stop to publish, and you can get fired from university quite easy if you don't have publications. So at the end, I found equilibrium in establishing a spin-off. So established spin-off that still is working that way. That's through the spin-off, we do the contractual research. So for spin-off, then I'm hiring people that, let's say, are not in threat of not very good publishing because they are technicians or people that simply I can keep with this money, including if they are not publishing, if they are somebody, this is not their cup of tea to do publishing, but they are perfect researchers, but simply they don't like this. So this is something that, of course, university has. This is a contract between spin-off and university. University has some shares from income or turnover of the company. So this is what is then paying the bills. And then, of course, we are now recently we are becoming finally successful in getting European grants, because of course Europe goes into crisis again. And fluctuations is not so visible because it's so scattered in Europe that the overall picture looks like that Europe is still fine, but not really. So more and more the grant agencies are not increasing the money, but the competition is bigger and bigger. So from the national schemes, it's not so easy to get the project and rather smaller projects. Yeah, everybody's telling, well, there is a lot of money in European Union. So we have European, it's called Horizon projects. And this is for big consortia. So you can have 15 partners in some project like that, but we just succeeded with one. So this is what is going to pay people. And we have another one smaller from the applied research agents in Czech Republic. So it's a combination. It's really a combination of the financial sources. It's permanent fight for money, because we don't have, as far as we are a research group, we don't have any money from teaching, including if we were doing teaching, we got no money. So I said, okay, so we cannot do teaching if we don't get money from that. So we are having all the PhD students that are basically coming with their own money. They are coming from some other universities, or we can support, of course, their research. But preferentially, they have some scholarship that can, at least for some time, cover their expenses better. Or like with Jonathan, if we have some positions for technicians, we are then taking these people as officially as technicians. Because you know how it is at university, you can have very nice budget, but you have the particular parts of the budget and you must spend only within these parts. So yeah.
Lukáš, I do have a question. So with the vertical farming movement and the possibility to go vertically in terms of analysis, do you see these technologies being more applicable in commercial facilities?
Absolutely, absolutely. This is why I'm here, because I wanted to learn a bit or to take a bit of understanding what is the CA. I mean, of course, we are in Europe. There is so far, of course, there are some facilities, but very difficult to get there. It would be interesting. I am trying to track, of course, the information coming from the money to these vertical farms. And I saw some presentations last time 1 year ago. There was a guy from R&D of Aerofarms speaking in Europe about their programs, trying to breed for flavors, etc., etc. We were speaking with Jonathan yesterday. I mean, this type of technologies, the affordable ones, are just perfect and enough to take all the germplasm, all the materials you have already, okay, tomatoes, strawberries, or whatever, and simply put them into the environment and to select what is growing best there. And for a long time, I'm also asking myself why people are still keeping in breeding and breeding and breeding to change some genotype to be more suitable for the environment in my region, when the environment has natural fluctuations and this fluctuation is also changing. So why we don't simply have some phenotypic data, maybe from barley from Argentina, and check, okay, now the climatology I have over the last years here is very much similar to this. So simply let's bring this variety to here, and maybe we have solved. We don't need to spend more money and time in breeding. You know, it's like a stubborn way. So what I'm trying to say is that if you know what is your environment, that you know perfectly because you are setting up in your facility, then probably it's only about the selection of the varieties or cultivars or whatever it's called to fit into that environment. And to do this screening, I believe you can use these technologies.
So two things on that. The first one is, I believe the vertical towers allow us to easily use imaging, because then you don't have a lot of structure in between, so you can move cameras up and down very easily. And actually, I've partnered with Georgia Tech, and there is a group of researchers already doing that. So the vertical system that is actually stacked is a little more difficult. And I do think that there is a lot of potential there in terms of using vertical systems in vertical farms. And also, we just hired the first CEA breeder for this particular goal of identifying better varieties that are more flavorful, that are better in color, and perhaps spending less time in the cultivar itself, but improving the environmental conditions for them. So there is also room for collaboration with this coming person that will join us soon.
I think that there is also one important thing, what we learned over the years, simple better. And yesterday I was speaking with boys about what I read, interview with ex-CEO of Fifth Season. That was vertical farming pioneer, or one of those that were really having more operations in more states of the US, and they just shut down the operations. And it was quite interesting because the guy was telling, investors were very interested in pumping money into this thing because, in automation, robotics, in plant growing or crop growing, that's cool. But they forgot the very important thing, that you are building facility to sell the crops. So they said, okay, automation for automation, don't do it. Try, and it would be our recommendation as well. Be simple. So for example, you don't need to probably have a lot of automation so far in the systems if you always have there a person walking through. So it could be that the person that is anyway moving, because I believe that in the everyday there is a process that you are controlling something. So it could be a person simply going with the telephone. And we were speaking about these simple things like make again, if you want to always take the picture of this thing, you take your telephone, and now, okay, what is the distance? So you simply make a stick, very simple thing. The stick is making your distance, take a picture to standardize somehow the picture. So it's about the standardization of the pictures to have easier life then. And we were speaking also about the lights yesterday, right? So if you have this blue, at the end, the purple light, this was, as you were telling, it was not allowing the people to control maturity of the fruits or controlling them. So I think that after the very big boom of the technologies for vertical farming, I think there will be like some time to rethink the thing and to try really to use the tools that can help you very efficiently only the moments that they can really help you, and same time are not giving you another complexity or kind of this. Yeah.
If you can add, we don't grow plants, or at least those farms, they should not worry about just growing plants, they should worry about selling plants.
And that is right.
Because if you give light, water, nutrients, CO2 and oxygen, you can grow a plant. That's no brainer.
I fully agree.
Thank you very much. I appreciate the time you put on presenting. Please feel free to reach out to Lukáš directly and to interact with our representative from the university during this next 4 weeks over there. It seems like we can build a lot of stuff while you're here. I will take your offer.
Let's keep it natural. Don't simply, there's something coming, let's do it. Don't force ourselves. We simply